Chapter 4, Tissues. The study of tissues is also called histology. Hist and tish are the same word. It comes from the Greek. So histology, study of tissues. So what are tissues? What they are groups of cells that are similar in origin, in structure, in function, that are working together to accomplish something. There are 4 fundamental tissue types. There are many subtypes of this, but everything falls into 4 categories. There's epithelial tissue, which is covering or glandular. It all derives from the same part of an embryo, etc., etc. There's muscle tissue, and muscle tissue is contractile with force, and it has certain properties that we will talk about. There's nervous tissue, which is an excitable tissue that is used to control the activity of other tissues and organs. And lastly, there is connective tissue, and really connective tissue is anything that's not epithelial, muscular, or nervous tissue. We'll just leave it at that. Connective tissue has certain properties, and we're going to talk about each of those properties. So, nervous tissue is in brain, spinal cord, nerves. Muscle tissue is attached to bone. Skeletal tissue, or the heart, cardiac, or the walls of hollow organs, which is smooth muscle. Epithelial form boundaries or secretes, and connective tissue supports, protects, and binds other things together. So we'll start out with epithelial tissue, and there's really 2 types of epithelia. There's covering and lining epithelia, and this really separates environments. It separates the inside from the outside. Your skin is what keeps the outside out and the inside in. Along your digestive tract, your reproductive tract, etc., the lining is called the mucosa, and that is made up of an epithelia. And within the body, epithelial line cavities and surround organs. So it's really keeping 2 distinct areas separate from one another. It's its main job. The second type of epithelium is called glandular epithelium, and they really are secretory cells. This is tissue that secretes things. So salivary glands would be epithelial in nature because they secrete saliva. The adrenal cortex is epithelial in nature because it secretes the corticosteroids. So what we're going to do is kind of have a couple of lists. One is the characteristics, what does tissue look like, and the other thing is what do we use them for. So this is the characteristics list. When you look at epithelial tissue under a microscope, when you look at some unknown tissues, like if it's on CSI, they look and they say, what is this? These are the characteristics they look for to tell if it's epithelial. So the first thing on the list is that all the cells in an epithelium have got the side that faces one environment and the side that faces the other environment. And both sides of those cells are different. So we say that the cells have polarity. They have an apical surface and a basal surface. The apical surface tends to be free. It is in the cavity or outside. The basal surface tends to be attached to the tissues that it's surrounding. The apical surface can have all kinds of modifications, things like microvilli, cilia, that kind of stuff. So if you see cilia, chances are good that it's an epithelial. That's the apical surface of an epithelium. Now, all of the basilar side is attached to a non-living layer called the basal lamina, and it's a glycoprotein. It's really glue, and it glues it down. If you think of a tabletop, like a desktop in one of the rooms that were at the college, the formica of that is epithelium. The particle board of it is connective tissue, and the basal lamina is the glue, the contact cement that holds the two of them together. If you think of a roof, the shingles are the epithelium. The wood sheeting that the shingles are nailed into is the connective tissue, and the nails are the basal lamina, hold it in place. Epithelial tissue is cellular in nature. It's closely packed cells, continuous sheets, and the cells are joined together with special junctions called desmosomes or tight junctions, things like that. We'll talk about those a little bit later. All epithelium is supported by connective tissue. There has to be something that supports it. Again, what good is a bunch of shingles if there's no wood supporting them? What good is that formica desktop if there's no particle board? What good is the paint on the wall if there's no drywall? Now, the cells have to be continuous, because that's what forms the barrier. They are really cell attached to cell attached to cell. Because the cells are so attached, there's no room for blood vessels. So you'll find that epithelial tissue is avascular. But the nerves end up in it, so it's innervated. It's a good place for nerves to be because it represents the boundary between two different environments. So if you want to monitor the environment, it's a good place to do it, is right in the epithelium. So that's why paper cuts don't bleed. If the paper cut only is in the epithelial layer of your skin, then it doesn't bleed. If it gets down into the connective layer of the skin, the dermis, then you get bleeding. So all of that supportive connective tissue is vascularized, and it's usually highly vascularized. So avascular and innervated. Another characteristic of epithelial cells is there's a high rate of regeneration. Because epithelial cells bear the brunt of changes in environment, they get damaged very easily. So they're constantly being replaced. They're constantly being worked on. So that means that there's a lot of mitosis. As you look at a sample of epithelial tissue, what you're going to end up seeing is quite a few of those cells. You're going to see lots of cells with one side, the free surface, looking different than the basilar surface. You're going to see connective tissue and no blood vessels in it, and you're going to see a lot of the nuclei in one of the phases of mitosis. You are going to see that. Now, before we get into this, I'll say this, that cancer cells are highly regenerative. They are in mitosis a lot. The target of chemotherapy and radiation is any cell that's in mitosis. That's why when people are undergoing cancer treatments, when they are having chemotherapy, they end up with problems of their epithelia. They end up with sores, and their hair falls out, and they have ulcerations and things like that, because epithelial cells are killed at the same time as tumor cells are killed. So in that covering epithelium, covering and lining epithelium, we really have to know, we have to subdivide it into a couple of things. So you ask yourself a couple of questions. Is it found in layers? If there's one layer, it's called the simple epithelium. If there's more than one layer, it is called the stratified epithelium. So simple epithelia only have one layer. The advantage to that is that pretty easy to get things through. So we use them in places where we're going to have a lot of absorption and that kind of thing. Stratified is more like shingles. You have these multiple layers. It's much harder to get absorption through a stratified epithelium. And the second question to ask yourself is, what type of cells are they? If the cell looks like a pancake or a fried egg, it's called a squamous cell. If the cell is the same in all dimensions, it's cuboid. And if the cell is tall and narrow, it is a column, it's columnar, so that it ends up looking like this. Squamous being the little flat fried egg one, the cuboid more or less the same in all directions, and columnar, tall, narrow ones. So if you put these things together, you can get a stratified squamous, you can have a simple columnar, you can have a stratified cuboid. All of these epithelia will fall into one of these characteristics. And these dictate the characteristics of that epithelium. And so we find them in very predictable places. So, and it's all together. You have the type of cell, the description of the cell, squamous, columnar, cuboidal, stratified, or simple, and that indicates the functions that they can happen there, and the functions that can happen there will then indicate the locations that we're at to find them. So when we go to something like a simple squamous epithelium, it is very thin, it's not very tough, so it's going to be mostly for exchange, things going in and going out. So diffusion, filtration, it really is a good place where things have to get across from one side to the other. So we see this in places like in the lungs. There's squamous epithelium, simple squamous epithelium in the alveoli of the lungs, the place where air exchange happens. We have them in lining of the blood vessels in the heart. It's called the endothelium. Really, when you think about it, blood is non-abrasive, and so you don't need a tough epithelium. You just need to keep it separated from the layers of the blood vessels or of the heart. So the same is true of the lining of your abdominal cavity. The serosa of your abdominal cavity would be a simple epithelium, because exchange happens there. You don't want those to be kept completely separate. You want fluids and things to change. The serosa of that body cavity, everybody will recognize if they've ever cleaned a fish. It's that very thin transparent tissue that makes the inside of the cavity glossy and makes the outside of the guts glossy. That's called serosa. It's a simple squamous epithelium. Simple cuboidal epithelium is going to be very much the same in that absorption will come across, but there will also be some secretion because there's now enough cytoplasm that there's enough organelles that these cells can actually make products that will get secreted. So we find these in the tubules of glands, on the surface of the ovary, in the tubules of the kidney. The kidney tubules in the nephron really are about absorption and secretion. So this is an important thing. So each one of these is a tubule, and each cell in the walls, and you can see that it is all cellular, surrounding the lumen here. There's an apical surface and a basilar surface. So that is a simple cuboidal epithelium. This kind of purple stuff here is the basal lamina and the connective tissue stroma that supports it. Simple columnar epithelium ends up looking like this. The cells tend to be elongated. Now, there's going to be some absorption because it is a simple epithelium. There's only one cell separating one environment from the other, so things will be able to go across. But there's going to be much more secretion than in a cuboidal. So this is mucus or enzymes or other things that are made and being secreted, usually out of the apical surface. They tend to have cilia on them. So because they tend to make mucus and cilia move mucus, they tend to have ciliated ones. So there's non-ciliated ones that are lining most of the digestive canal because they're secreting enzymes and things like that. The ciliated ones are like in the lungs, in the uterus, uterine tubes, that kind of thing. So still absorption and more secretion than the cuboidal because there is more cytoplasm, there's more opportunity. And these are the ones that you tend to see cilia in. Now, there is a variation on this where some of the cells are small, almost cuboidal in shape, and some of them are tall and columnar in shape. But they all reach the basilar surface. Not all of them reach the apical surface. And we call this pseudostratified. So they tend to have cilia, but you can get away with them not. And you can see in this photograph that these cells are the cells that produce mucus. So there's a lot of mucus-producing cells happen in pseudostratified columnar epithelium. Remember, it's pseudostratified, which makes it simple, a simple epithelium. We see this in the trachea and the upper respiratory tract, places like that. The lumen on this side tends to have air. When we get into the stratified ones, a very common one is stratified squamous epithelium. So skin is a stratified squamous epithelium. It doesn't work for absorption. Its function is to protect from especially abrasion, so friction. So that's why it's on the skin. The surface cells may in fact even be dead and just stuck on there before they are rubbed off. And so there's there tends to be layers. The layers closest to the basement membrane are the ones that are undergoing mitosis. The layers closest to the surface are almost sacrificial. They're the ones that are taking the most abuse. So there's a couple of different types. In skin, there's a protein in it called keratin. So that would be a keratinized stratified squamous epithelium. That would be the epidermis of your skin. There's non-keratinized versions where it stays soft. So this is the moist linings of your digestive system, your respiratory system, and your reproductive system. So so this would be like inside of your mouth, down your throat, the esophagus has this kind. So once it gets past the stomach, it doesn't, because secretion and absorption are more important. In chewing and swallowing, there is a fair amount of abrasion. You have to protect against that. So it tends to be non-keratinized stratified squamous epithelium. The vagina has this as well, for obvious reasons. As the birth canal, it's subject to a lot of force, and just in intercourse, it is subject to abrasion and force. The problem with the non-keratinized stratified squamous epithelium is there's no room for mucus-producing cells. So it is a dry epithelium unless it can be moistened from an external source, from a glandular source that's remote from it. There are stratified cuboidal. In mammary glands, some sweat glands have it. It's not very common. It doesn't end up with multiple layers. It's usually 2 layers. And its job would be mainly one of secretion. So it would be glandular secretion. And then the stratified columnar epithelium is even more limited. So you get some in the pharynx, some in the male urethra, because the male urethra has to carry both reproductive functions and urinary functions. So basically it has to get replaced quickly, but it also needs a lot of mucus, and it needs a lot of secretory things going on. So so it can happen. Oftentimes what ends up happening is that this is found in transitional epithelium, in the change between one type of epithelium to another. So for instance, the cervix in the female reproductive system would be a columnar epithelium, either pseudostratified or a genuine columnar epithelium. The vagina is a stratified squamous epithelium. Around the edge of the cervix, it will be cells that are kind of stratifying, but turning in more into columnar. So they're not stratified squamous. You'll have some stratified cuboidal, and then some stratified columnar before you get into the straight kind of pseudo-stratified columnar epithelium of the cervix itself that's producing a lot of mucus. These are the cells that they are sampling when they do a pap smear, and they're looking for abnormalities that would indicate inflammation and morphological changes in that transitional epithelium. And that doesn't necessarily mean bad things, it just means that it's worth looking at. I had a surgeon say to me about some polyps in my intestine during a colonoscopy, or right after his colonoscopy. He said, not all polyps turn into cancer, but all cancer starts as polyps, so it's best to deal with it. So this is a picture of some transitional epithelium, and it's really hard to pinpoint what it is. So the glandular epithelium is cells that secrete. Now, we classify them, glands, as to whether they secrete into the body or outside of the body. Glands that secrete into the body are called endocrine, and glands that secrete outside of the body are called exocrine. Crine means to secrete, so endo and exo, in and out. We also can talk about the number of cells that form the gland. So mucous cells are exocrine glands that are unicellular. Now, remember, the lumen of your digestive system is outside the body. I will do that in class. I will do a demonstration if somebody reminds me. So, endocrine glands, because they are already within the body, they don't need a duct. They don't need to carry their secretions outside of the body. So there's no duct. So they are often they are ductless glands. And so they secrete things right into the interstitial fluid, which ends up in the blood or the lymph, and gets carried to its target organs that are also within the body. They tend to make things that are called hormones. And actually next semester we'll be doing a whole chapter on the endocrine system and hormones. Exocrine glands are everywhere. So these are sweat glands, these are mucus glands, these are salivary glands, these are sebaceous glands that secrete oil, these are the digestive enzyme glands that in the pancreas that secrete digestive enzymes that mix with the food. These are the hydrochloric acid producing glands in the stomach that produce stomach acid. These are the pepsinogen producing ones that produce enzymes that break down proteins in the stomach. All of those are exocrine glands because they secrete outside. They tend to have ducts. Not all of them do. Mucus glands don't necessarily have ducts, but they tend to have ducts. So the goblet cells, the mucus cells, are the only ones that really are ductless, and they're the only important unicellular gland. And they end up looking like this. So within a columnar epithelium, there's a nucleus, there's a bunch of Golgi apparatus and endoplasmic reticulum that's making this protein called mucin that will erupt out there and secrete these vesicles that contain the mucin. When the mucin mixes with water, it becomes mucus. So the multicellular exocrine glands have ducts and can be classified by the duct type. Whether there's one duct, it's a simple. If the duct is branched, it's compound. The secretory cells also can be arranged like test tubes, so they'd be tubular, or like berries, alveolar. Alveolus means berry in Latin, so if they're kind of berry shaped, so they look like grapes, they would be alveolar. Or tubular alveolar, so it's a mixture of both. So that we end up with things like this. So simple ducts, so there's one duct here that comes off these. So this would be a simple tubular one. This would be a simple alveolar one. This would be a simple branched tubular. See, there's three tubes, but there's only one duct here. Simple branched alveolar. So this would be a sebaceous gland. Some stomach glands are like this. So oftentimes what can happen is that different parts of different cells here will be secreting different things, and what ends up being secreted out there is a mixture of things. This one's called a compound tubular. So all of the secretory units are test tube shaped, they're tubular, but you'll see that here there are three distinct ducts coming together to the one pore. Compound alveolar is the same, three distinct, but with alveolar shaped secretory. Compound tubular alveolar is a mix of them. These ones especially probably have these cells secreting something entirely different than these cells, and etc. So salivary glands and that kind of thing would be a common case of that. So all the different things that are found in saliva end up mixing together as they come out this long salivary gland duct. And there's lots of branches. So how do these cells actually secrete? Again, there's three ways of doing this. One is called merocrine. So merocrine is secretion by exocytosis. So what happens is that the Golgi apparatus packages little vesicles, and the vesicles release the contents a little bit at a time. So we use merocrine where we are more or less getting a constant outflow of the secretions. Like as fast as the cells can make it, they're secreting it, and they don't tend to hold back on it. Holocrine cells, on the other hand, what happens to them is that the proteins, the products that are built in the cell, build up and build up and build up until the cell actually ruptures and the contents leave in a big burst. Everything leaves at the same time. But the cell is done. The cell is dead when that happens. So it has to be replaced. Holocrine secretions tend to have more lipids in it because there's cellular debris, there's cell membranes and things like that. So sebaceous glands are typical holocrine. Well, that's the same picture. I don't know why that is there. So the last type is called apicrine. Now it's not listed here because there's a controversy as to whether apicrine, which is the apex, breaks off, and so it's like one large vesicle. So it's kind of halfway between holocrine and merocrine, in that the top half of the cell ruptures, but the bottom half of the cell with the nucleus and the Golgi apparatus and everything stays intact. There is controversy as to whether it even exists in the human body. When I was in school, we considered that mammary glands producing milk was done this way, and now they're deciding that it is probably more merocrine with one large vesicle. It is one of those kind of almost pointless debates. That's about it for epithelial tissue.